All right, continuing on with the Materialist Metaphysics of Gilles Deleuze from Manuel de Landa's book, a collection of essays entitled Deleuze, History and Science. Please listen to the previous video so this will make sense to you. I'll just start in here. The overall metaphysical picture that emerges from these considerations is quite different from the Aristotelian one. In the latter, the world is already segmented by logical categories, some more specific, others more generic, pre-existing segments that act as formal causes to generate all the particular members of each category. For Deleuze, on the other hand, the world is the first and foremost a continuum of intensity that becomes segmented into species only as certain tendencies are manifested and certain capacities exercised. In the case of atomic assemblages, the intensive continuum is embodied in stars, balls of plasma possessing a minimal segmentation, but not entirely undifferentiated, since they have a structure defined by differences of temperature, pressure, and density. The possible ways of segmenting this continuum are not given by a logical subdivision of a genus into species, but by a virtual structure that can be captured mathematically. At this point, Aristotle could raise an objection. This neo-materialist metaphysics derives most of its intuitions from physics and mathematics, but the whole point of creating a metaphysical science was precisely to discover speculatively those aspects of the world that are prior to both the physical and the mathematical. To this we may respond that the use of results from physics and mathematics does not imply that metaphysics can be subordinated to either one, not that it should not be satisfied with a priori speculation. Only that it should not be satisfied with a priori speculation. In a Deleuzian ontology, the main philosophical task is to extract from the results produced by physicists and mathematicians a metaphysical problem. Why is the production and maintenance of atomic identity the way that it is? So essentially that is in a nutshell, the question of the Deleuzian ontology is the appropriation of the insights provided by physics and mathematicians to create a metaphysical problem, which is why is the production and maintenance of atomic identity the way it is? A careless physicist might, for example, pose this problem incorrectly, asserting that atoms are what they are because of the immutable laws of nature. But this would hardly take us beyond Aristotle, since it simply replaces one set of formal causes, general categories, with another one, general laws. Worse yet, most physicists espouse a positivist ontology, that is, they are committed to assert the mind-independent existence only of that which is directly observable. This means that they would not refer to temperature and pressure, for example, as emergent intensive properties of stars, but as quantities, that is, as quantities that are directly observable when reading a thermometer or a barometer. Similarly, when using the term law, positivists would not refer to the immanent patterns of being and becoming to which the term refers in a realist ontology. Instead, they would use the term to refer to the equations used to model these patterns, because equations are directly observable when written on a blackboard or printed on a piece of paper. Clearly, a positivist ontology makes it impossible for physicists to correctly pose metaphysical problems of existence, and similarly for mathematicians espousing a Platonist ontology. But what about the objection that the content of science is constantly changing and that building a metaphysics on top of it is like building a house on a foundation of sand? It is true that the details of the causal mechanisms involved in a particular production process may change when new empirical findings determine that they were wrong, or when they are improved upon. And similarly, for the mathematical ideas used to conceptualize the mechanism-independent structure of possibility spaces, in the future, new advances in mathematics may reveal novel connections between the relevant fields, dynamical systems, group theory, or even generate entirely new fields. These innovations may indeed provide better tools to conceptualize universal singularities, much as novel empirical discoveries may provide new resources to conceptualize the process that produce individual singularities. Metaphysics must remain sensitive to these potential changes and remain capable of profiting from them. Nevertheless, precautions must be taken. We must make an effort, for example, to use only scientific results that have withstood the test of time, not the empirical findings of cutting-edge science. It would be unwise to speculate about the problem of existence using what we know today about esoteric entities like dark matter or cosmic strings. But who could seriously doubt the existence of hydrogen or oxygen atoms? Although all scientific results are supposed to be falsifiable, this only implies that no result should be established a priori, not that all facts are equally likely to be false. To falsify the assertion that hydrogen or oxygen atoms exist, for instance, we would have to simultaneously falsify hundreds of other statements, like the statement that a water molecule is composed of 2 hydrogen atoms and 1 oxygen atom. But can any realist philosopher seriously consider such a wholesale falsification possible? It is crucial not to let metaphysical speculation be constrained by particular theories of science, especially theories taking as their object of study the reified generality, science. In a neo-materialist metaphysics, there is no such thing as science in general, only a population of individual scientific fields, a population that is not converging on a final truth, but rather growing and diverging as it tracks a reality that is itself divergent. These remarks should be kept in mind as we attempt to eliminate other transcendent entities, in particular, replacing Aristotle's own example of genus, animal, and the species horse or human, involves using results from a variety of biological fields, some of which, paleontology, evolutionary biology, are older and better established than others, genetics and embryology. Let's begin with a description of the results from the safest fields. Today, it is widely accepted that biological species are as singular, as unique, and as historically contingent as individual organisms. Species are born when their gene pool is closed to external flows of genetic materials through reproductive isolation, and they die through extinction. In other words, they belong to the category of entities that Aristotle regarded as incapable of yielding metaphysical knowledge, since, as he wrote, things that are subject to corruption and decay are obscure to those even that are in possession of scientific knowledge. Similarly, the defining properties of a biological species are not necessary as Aristotle would require. Reproductive isolation is a contingent achievement that varies by degree, so nothing guarantees that the identity of a biological species will endure forever. And since the anatomical, physiological, and behavioral properties of organisms of a given species are produced by historical processes that cannot be exactly duplicated, driving a species to extinction is like killing an individual organism, that is, eliminating an entity that can never return again. Clearly, the evolutionary conception of species is quite different from the Aristotelian one. The relation between organisms and species is not one of membership in a general category, but one of reproductively interacting parts composing an emergent whole. In other words, species are nothing but assemblages of organisms having the same ontological status, individual singularities, but operating at a larger spatio-temporal scale. A common history in which the ancestors of the organisms that compose a new species faced similar challenges from predators and parasites. Scarce resources and climatic changes is what produces the bodily resemblance that we use to classify them. Shared selection pressures homogenize their gene pool, allowing us to infer that these organisms have more genes in common with each other than with organisms of other species. But as in the case of protons and neutrons, we need to stress not only what stays the same, but also what varies. Without a constant production of genetic differences by accidental mutations or sexual recombinations, selection pressures would have no raw material to operate on, no low fitness variants to filter out, or high fitness variants to promote. Let's now describe the intensive continuum that is progressively segmented as new species emerge. As in the case of stars, that term continuum does not imply an absolute absence of segmentation. Stars may not be segmented by chemical species, except those that they themselves produce and burn as fuel, but they certainly are segmented by subatomic particles. Similarly, when speaking of an ecological continuum, the claim cannot be said that it was not originally segmented physically or chemically, only that it was unsegmented biologically. Roughly, the first discrete biological segment to emerge consisted of flat layers of motionless bacteria inhabiting the interface between ocean water and bottom sediment. The earliest bacteria fueled themselves by fermenting available minerals, but they eventually evolved by the capacity to tap into solar radiation through photosynthesis, greatly enriching the intensive continuum. The term intensive refers to physical properties like temperature or pressure that can form gradients containing useful energy. A gradient of temperature is simply the result of coupling hot and cold masses of air or water, while gradient of pressure results from coupling high and low pressure masses. Chemical gradients, in turn, can be created by coupling materials with different pH, some acid, some alkaline, or materials with different properties of reduction and oxidation. These coupled differences in intensity have the ability to drive processes, an ability that had to be harnessed by the earliest living creatures if they were to survive, proliferate, and evolve. And once primitive bacteria developed the means to tap into physical and chemical gradients, they themselves became a biological gradient, a concentration of biomass for the ancestors of today's amoebas and paramecium, that is, for the unicellular organisms that preyed on them. These primordial food chains possessed only a few distinct segments, but as they complexified, a much more thorough segmentation became possible as new ecological niches opened up and new species came into being to occupy these niches. Today, solar energy and mineral nutrients are encapsulated into the bodies of countless animals and plants of different species, a highly segmented condition when compared to the characterizing the earliest biosphere. Having replaced Aristotelian species like horse and human, defined by a set of necessary and timeless characteristics, the next move is to identify the structure of possibility spaces that replaces the genus animal. Unfortunately, unlike the fields of evolutionary biology and ecology that provide relatively solid results ripe for metaphysical speculation, the biological disciplines on which we must rely for this task are at the cutting edge of research. Their results are therefore much more likely to change in the near future. As one embryologist suggests, the relevant results are today closer to the ontological status of dark matter than that of hydrogen or oxygen. This means that we have to break the rule of using only results that have endured the test of time. Metaphysically, what we need to conceive is, in the words of Deleuze and Guattari, quote, a single abstract animal for all the assemblages that effectuate it. For the vertebra to become an octopus or a cuttlefish, all it would have to do is fold itself into fast enough to fuse the elements of the halves of its back together, then bring its pelvis up to the nape of the neck and gather its limbs together into one of its extremities. End quote. In other words, we need to conceive of a topological animal that can be folded and stretched into the multitude of different animal species that populate the world. Deleuze and Guattari do not, of course, believe that these topological operations can be performed on adult animals. Only the embryos of those animals are flexible enough to endure these transformations. Moreover, the topological or virtual animal must be capable not only of being divergently actualized into many different assemblages, but each actualization must be inheritable. That means that the possibility space we are searching for is not just one of possible animal forms, but also of possible genetic combinations that can reliably and recurrently produce those forms. There is one more point that must be clarified before we begin to describe these possibility spaces. While the Aristotelian classification, there are only two levels. In modern taxonomics, there are many levels, with species and genera at the bottom and kingdoms and phyla at the top. Aristotle's animal genus is today animal kingdom, subdivided into different phyla, chordata, arthropoda, in turn subdivided into subphyla like vertebrates and insects. Unlike the lower taxa, the higher ones are defined by a common body plan, the features of which are often defined early in embryological development. It is the structure of the space of possible body plans that replaces the genus animal. The first problem we must confront is that the possibility spaces involved are very different from the ones in which we have over a century of experience, like phase space and its topological singularities. Unlike phase space, which is fully continuous and has well-defined spatial structure, the space of possible genes is entirely discrete and has no intrinsic spatial order. In addition, what is singular, special, or remarkable, let alone universal, in the structure of discrete and combinatorial spaces is not well understood. So we are in uncharted waters here and must proceed carefully. Let's begin with what we do know. The genetic code itself is by now well established and used routinely in industry to create proteins out of genes, or to create a designer gene from a natural protein. Both genes and proteins are linear sequences of molecules differing only in their components, nucleotide molecules in the case of genes, amino acids in the case of proteins. The genetic code is simply a way of mapping one type of molecular sequence into another, 3 nucleotides corresponding to each amino acid, the correspondence itself being arbitrary, a kind of frozen evolutionary accident. The one thing we know for sure about the spaces of possible genes and proteins is their enormous size. The number of possible sequences of a given length is the number of available components raised to the maximum length. Genes are composed of only 4 possible nucleotides, while proteins can be drawn from a repertoire of 20 possible amino acids. A very short protein, 5 amino acids long, can exist in over 3 million different combinations, the number 20 raised to the 5th power. The number of possible proteins, 300 amino acids long, the average length of a contemporary enzyme, is literally infinite, larger than any number of seconds to the universe has existed. And similarly for genes. Although in this case, the number of different components is much smaller, their length is 3 times greater, since they must use 3 nucleotides to specify a single amino acid. Thus, in either case, we are considering combinatorial spaces that grow explosively as the length of the sequences increases. But how can we study the structure of these infinite spaces if they lack any intrinsic spatial order? One strategy would be to impose on them a non-arbitrary order, that is, an order that has some connection to the tendencies and capacities of the molecular sequence in question. In the case of genes, the capacity to replicate is crucial, as is the tendency for copying errors, mutations, to occur during replication. So a reasonable spatial order can be imposed if we arrange each sequence of nucleotides so that it has neighbors, all sequence that differ from it, form it by only one mutation. The resulting space is multidimensional because it must include all the variants that can be created by varying each nucleotide along the full length of a given gene, and the resulting one mutant variant must be assigned a different dimension. But while the sheer number of dimensions make the space very complex, it also greatly simplifies the distribution of sequences after we rearrange them every possible gene is in direct contact with all its one mutant neighbors, forming a connected path that evolution can follow. In other words, given this spatial arrangement, genetic evolution can be visualized as a continuous walk from one neighbor to the next, driven by events producing one mutation at a time.